For decades, scientists thought that roughly half the cells in your brain were essentially just useless bubble wrap.
Yeah, literally just packing material.
Right. I mean, early anatomists looked at these cells, noticed they didn't fire electrical signals like neurons did, and just assumed they were there to keep the important stuff structurally secure. They even named them ganglia, which literally translates from Greek to glue.
Just brain glue.
Exactly, just brain glue. Today, however, we know that if those so-called bubble wrap cells fail, you might develop schizophrenia or bipolar disorder or even severe treatment-resistant depression.
It's honestly one of the greatest scientific paradigm shifts of the last century. I mean, we spent so much time looking at the neurons that we completely ignored the environment they lived in.
Mm-hmm.
And, understanding that completely changes how we view mental illness today.
It really does. So welcome to this deep dive. Today we're exploring the architecture of thought itself. We're acting as your companion to Kaplan and Sadok's comprehensive textbook of psychiatry, specifically Chapter 1.2 on functional neuroanatomy.
Brilliant chapter.
It is. And to really grasp this, I want you to picture a map of a major city, let's say London. When you look at that map, you don't just see a static piece of geography, right?
No, of course not.
You see a bustling metropolis. You see zones of commerce, residential neighborhoods, transport hubs, all connected by this incredibly intricate web of roads, rails, and communication lines.
Right. It's alive.
Exactly.
Yeah.
A city isn't defined just by its buildings, but by the movement and the organized conversation happening between them. So if neuroscience asks how the brain works, functional neuroanatomy asks where those processes unfold, and most importantly, how meaning emerges from their connections.
I absolutely love the city map analogy because it forces us out of the old way of thinking.
Phrenology stuff.
Yeah, well, historically, there was this intense focus on strict localization, the idea that specific, highly complex functions lived inside discrete, isolated islands of the brain. But what we know now is that localization is really only a tiny part of the story. I mean, while it's true that certain functions cluster in specific regions, complex psychiatric phenomena arise from circuits.
Networks, essentially.
Exactly. They arise from networks. We're not here today to just memorize a list of Latin terms. The goal is to build mental models. We want to clarify the mechanisms of the brain, see how they manifest in human behavior, and ultimately, strengthen your clinical reasoning.
And that brings us to the core anchor for our entire discussion today, and really for your entire clinical practice. Psychiatry is the study of how brain, mind, and experience interact, and how that process can go wrong.
Spot on.
So to start unpacking this, let me pose a question that I think a lot of people secretly wonder about when they look at modern medical tech.
Okay, go ahead.
We have mapped the whole human brain, right?
Okay.
We have functional MRI, we have diffusion tensor imaging. So why can't we just point to the depression center or the schizophrenia center on a scan and say, hey, there it is, that's what's broken?
Well, simply because that center doesn't exist. Let's take emotion as an example. Emotion isn't just sitting in the amygdala waiting to be triggered.
Right, it's not a button you press.
Exactly. Emotion is a continuous, highly dynamic conversation between the amygdala, the prefrontal cortex, the hippocampus, and the brainstem.
Wow, so it's a whole committee.
It really is. When you experience fear, your amygdala is sounding the alarm, your hippocampus is checking to see if you've seen this threat before, your prefrontal cortex is trying to logically evaluate if you're actually in danger, and your brainstem is cranking up your heart rate.
All at the same time.
All at once. So if someone has an anxiety disorder, it's rarely because one single structure is physically broken. It's because the communication lines between them are dysregulated. The metropolis is in gridlock, basically.
So it's less about a single demolished building in our city and more about a subtle breakdown in the transport network, or maybe the power grid connecting those buildings. Okay, let's unpack this by starting at the very foundation of the city, the cellular level. The bricks and mortar. We all know about neurons. I mean, the human brain has roughly 100 billion of them.
Give or take. Yeah.
You have your cell body, the dendrites receiving the signals like tree branches, the long axon sending the electrical signal down the line, and the axon terminals releasing chemical neurotransmitters across the gap, the synapse, to the next cell.
Right. And within those 100 billion neurons, we broadly categorize them into two types. You have projection neurons, which have these incredibly long axons that communicate across vast distances, say from the cerebral cortex all the way down to the spinal cord.
So those are the highways.
Exactly. And then you have local circuit neurons, often called interneurons, which have very short axons and process information locally, sort of like middle managers in an office.
That makes sense.
But as you alluded to in the introduction, if we only talk about neurons, we're missing a massive, arguably more fascinating part of the story.
The bubble wrap. The glial cells.
Yeah. Exactly. The ganglia. This view that they are just structural support has been entirely overturned. Glial cells are critical, active participants in brain function. And there's one specific type we need to focus heavily on, which is the astrocyte.
Okay. Astrocytes.
Astrocytes come in two main varieties. You have fibrous astrocytes, which are found mostly in the white matter, and they do provide a lot of that structural support. But the protoplasmic astrocytes, which live in the gray matter, are just an absolute marvel.
All the bushy ones, right?
Yeah, they look like incredibly dense, bushy stars, and they have these elaborate processes that interdigitate. They literally weave themselves between neurons and blood vessels.
I've actually heard protoplasmic astrocytes described as the city's power grid managers. Let's walk through that mechanism because they literally form what's called a neurovascular bridge, right?
They do. Their specialized physical extensions, which we call end-feet, literally wrap around the tiny capillary blood vessels in the brain. They act as the middleman.
Pulling resources out.
Exactly. They extract glucose directly from the bloodstream, but they don't just hand it over to the neuron. They convert that glucose into a substance called lactate and then feed that lactate to the neurons.
Wait, why lactate?
Well, neurons actually prefer lactate as an energy substrate when they're firing rapidly. So the astrocyte is actively managing the energy supply chain based on exactly how hard the local neurons are working.
That's incredible. It's not just passive diffusion, it's an active metabolic shuttle.
Mm.
But it goes far beyond just providing power, doesn't it? Because protoplasmic astrocytes are deeply involved in neurotransmission itself.
They are, yes.
And this brings us to a concept that completely blew my mind, the tripartite synapse. I mean, in medical school, you're taught that a synapse has two parts, you know, the presynaptic neuron sending the signal and the postsynaptic neuron receiving it. Two parts.
The classic view, yeah.
But the astrocyte is the third partner.
Yes, the tripartite synapse. Yeah. The parasynaptic processes of the astrocyte essentially wrap around the synaptic cleft, the gap between the two communicating neurons.
You're right.
When the presynaptic neuron releases neurotransmitters like glutamate, which is the brain's main excitatory signal, or GABA, which is the main inhibitory signal, those transmitters don't just hit the receiving neuron, they spill over and stimulate receptors on the astrocyte as well.
Wait, so the astrocyte is literally listening to the conversation between the neurons?
Not only is it listening, it's actively participating. When the astrocyte detects glutamate, it can release its own chemical messengers called gliotransmitters to modulate the activity of both neurons.
Oh, wow.
But perhaps its most vital job is cleanup. Glutamate is highly excitatory. If it just sits in the synaptic cleft, it will continue to stimulate the receiving neuron endlessly.
And that leads to excitotoxicity, right? I want to make sure we define that clearly because it sounds like a sci-fi weapon. What actually happens to a cell during excitotoxicity?
It's a great term to clarify. Imagine turning the volume on a speaker up so intensely high that the physical cone of the speaker actually tears and destroys itself.
Ouch, okay.
That is essentially excitotoxicity. The neuron is stimulated so relentlessly by excess glutamate that its internal calcium levels skyrocket, which triggers a cascade of enzymes that literally digest the cell from the inside out. The neuron burns itself out and dies.
So the astrocyte is like the bouncer pulling the overexcited patrons out of the club before they wreck the place.
Exactly.
It vacuums up the excess glutamate and GABA from the cleft to prevent that toxic burnout, and it also buffers extracellular potassium to keep the electrical environment stable. It's actively shaping the flow of information.
So when we move from mechanism to clinical meaning, we have to ask, well, what happens if these power grid managers start failing?
The whole city suffers.
Precisely. And this isn't just theoretical. When we look at postmortem brain tissue from individuals with psychiatric disorders, we see striking alterations in the glia.
Like in schizophrenia.
Yes. In schizophrenia, stereologic studies, which simply means using 3D mathematical modeling to accurately count the total number of cells in a tissue sample rather than just guessing from a flat 2D slice. These studies show significantly altered glial cell numbers in the dorsal prefrontal cortex.
And we see it in mood disorders too, don't we?
We do. In major depressive disorder, we see a stark reduction in glial cell density, specifically in the subgenual medial prefrontal cortex, which is an area heavily involved in mood regulation.
That's fascinating.
Furthermore, functional imaging often shows a decrease in cerebral blood flow in the prefrontal cortex of chronically depressed patients. Historically, we thought the neurons were just inactive.
Right, just sort of sleeping on the job.
Yeah, but now we suspect that decrease in blood flow might actually be linked to a physical reduction in blood vessel coverage by those astrocytic end feet. The neurovascular bridge is literally crumbling.
Wow. So the neurons might be structurally fine, but they're literally starving because the astrocytes aren't delivering the lactate.
Yeah.
Now, before we move on from astrocytes, we have to talk about taking out the cellular garbage. Because if these cells are managing all this energy and neurotransmitter traffic, they must be generating a massive amount of metabolic waste.
Oh, absolutely.
How does the brain clean that up?
Yeah.
I want to bring in the glymphatic system here, because every single time I look at a psychiatric disorder, whether it's depression, bipolar, schizophrenia, PTSD, sleep disturbance, is always a massive comorbidity.
They go hand in hand almost universally. The glymphatic system is essentially the brain's macroscopic waste clearance pathway, and it relies heavily on the structural network of, you guessed it, astrocytes.
Right.
The cerebrospinal fluid flushes through the brain tissue, sweeping away metabolic waste proteins, and it travels along the pathways created by the astrocytic end feet. But here is the crucial part. This system is primarily active during duck sleep.
So during the day, the brain is accumulating waste, and at night, the glymphatic system opens the floodgates to wash it away.
Exactly. So think about the clinical implication. If a patient's sleep architecture is chronically disrupted, which is a hallmark of nearly all severe psychiatric conditions, the clearance of neuronal and glial waste products is severely impaired.
It's just built up.
Over time, this cellular debris builds up and alters the extracellular environment. This toxic buildup can directly impair neurotransmission, potentially exacerbating the underlying psychiatric condition.
It creates a vicious cycle.
Exactly. The illness disrupts sleep, the lack of sleep prevents waste clearance, and the waste buildup worsens the illness.
Okay, so that covers the astrocytes, our power grid managers, and waste disposal facilitators. But there's another type of glial cell that has also had a major rebranding recently, the microglia.
Ah, yes, microglia.
Historically, they were seen as just the cleanup crew, right? The scavengers that show up to eat dead cells after a stroke or a traumatic brain injury. But they are so much more than that. I like to think of them as the city's gardeners.
Gardeners is the perfect way to conceptualize them. Microglia are actually derived from macrophages, the white blood cells that eat pathogens in your immune system, but they migrate to the brain very early in embryonic development.
Okay.
In a healthy, uninjured brain, they aren't just passively waiting around for a stroke to happen. They are incredibly active. They constantly extend and retract their tiny physical processes, touching and monitoring the status of billions of synaptic connections.
And this ties into synaptic pruning, right? When a human is born, and especially as they move into childhood and adolescence, the brain produces a massive excess of synaptic connections. It's basically a jungle.
Right.
But you don't want a jungle. You want an efficient garden.
Exactly. Microglia are the primary agents of synaptic pruning. They actively evaluate which synapses are being used and which are redundant.
And then what?
And then they phagocytose, which is just a fancy biological term for swallowing and digesting. They phagocytose the dendritic spines that aren't pulling their weight. They physically chew away the unused connections, sculpting and refining the neural circuits to make them more efficient.
This pruning process is absolutely critical. I mean, if the gardeners prune too much or too little, the city's communication lines get completely tangled.
Precisely.
Well, let's use a chainsaw analogy here. If the microglia are given chainsaws instead of delicate pruning shears, they might start aggressively cutting down healthy branches until the neural tree is bare and can't receive any signals.
That's a very vivid but accurate way to picture it.
We'll come back to how exactly that manifests in conditions like schizophrenia in just a moment. But before we leave this cellular overview, I want to take a step back and ask a somewhat provocative question about how we study all this.
Okay, fire away.
Because we use animal models constantly, right? We test new antipsychotics on mice and rats. We look at their brains. But humans aren't just big mice. There is a massive evolutionary jump, isn't there?
A profound jump, and one that psychiatry has to grapple with constantly. Grossly, the most obvious difference when you look at a human brain versus an animal brain is the sheer size of the prefrontal cortex.
Right.
In a cat, The prefrontal cortex makes up about 3.5% of the total cortical volume. In a macaque monkey, it's about 11.5%. In a human, the prefrontal cortex occupies nearly 30% of the entire cortical volume.
30%?
Yes. That is a staggering evolutionary expansion of the area, entirely devoted to higher cognitive functions, abstract thought, social reasoning, and emotional regulation.
But I'm assuming it's not just size. I mean, if you just inflated a mouse brain to the size of a melon, it still wouldn't be a human brain. The actual cellular makeup has to be distinct.
You're exactly right. It's highly distinct molecularly and cellularly. Let's go back to our astrocytes. The ratio of astrocytes to neurons is about 5 times greater in humans compared to rodents. So we have significantly more glial regulation per neuron. Human protoplasmic astrocytes are also physically much larger, vastly more complex, and they reach exponentially more synapses. We even have entirely specific subtypes like intralaminar and polarized astrocytes that simply do not exist in the rodent brain.
Wait, really? We have whole categories of cells that mice just flat out don't possess.
We do. This enhanced astrocytic network strongly suggests that our uniquely human higher-order cognitive functions rely heavily on this complex glial regulation.
That is wild.
Furthermore, look at our inhibitory neurons. In the rodent cerebral cortex, about 15% of neurons are GABAergic interneurons, the cells that put the brakes on electrical signals. In humans, that number jumps to between 20 and 25%. We have a much more robust, highly complex system of cortical inhibition.
I have to push back on the research implications of this then, because this raises such a critical question about the limits of animal models.
It really does.
If we are testing a drug for schizophrenia, which is a uniquely human disorder characterized by highly complex delusions, paranoia, and abstract thought disorders, and we're testing it on a mouse that literally lacks the astrocyte subtypes and the dense inhibitory networks we rely on, aren't we basically testing a jet engine part on a bicycle? We have to be incredibly careful about the conclusions we draw, don't we?
It is a fundamental limitation of psychiatric research, and it's something the field is acutely aware of. Animal models are excellent. In fact, they are indispensable for studying basic molecular pathways.
Like receptor binding?
Exactly. If you want to know if a drug blocks a specific dopamine receptor, a mouse will tell you. If you want to study overt motor behaviors, animal models work. But when it comes to the highly nuanced, abstract circuitry of human psychiatric disorders, they often fall entirely short.
Because a mouse can't tell you it's hallucinating.
Right. A mouse cannot tell you it's experiencing a delusion of grandiosity. This is why human postmortem brain studies, alongside advanced functional imaging of living human patients, are so utterly irreplaceable in functional neuroanatomy.
Okay, that makes total sense. So let's say we have a human postmortem tissue sample. When neuroanatomists are looking at this tissue under a microscope, how do they actually make sense of it? Because if you just slice a piece of brain, it basically looks like a uniform grayish-white blob to the naked eye.
It does, yes.
To see the metropolis, you need to look at the architecture. And in the textbook, there are 3 main types of clues or architectural maps we use: cytoarchitecture, myeloarchitecture, and chemoarchitecture. Can you break these down? What are we actually looking at?
Certainly. Cytoarchitecture is essentially taking a census of the cell bodies. We reveal it using something called a Nissl stain. This is a specific chemical dye that binds to ribonucleotides in the cell body of the neuron.
So it ignores the branches.
Exactly. It ignores the long axons and dendrites and just stains the core of the cell. This allows us to see the size, the shape, and the specific packing density of neurons in any given area. When you look at a Nissl stain of the cerebral cortex, you don't see a uniform blob. You see 6 beautifully distinct horizontal layers of cells, each with different densities.
Okay, so cytoarchitecture is the cell bodies. What about myeloarchitecture?
Myeloarchitecture maps the wiring. It uses silver stains to specifically highlight the myelin sheaths, the fatty insulation that wraps around the long axons. This reveals the dense white matter tracks, showing us exactly how different regions of the brain are connected. It's the roadmap of the highways.
Brilliant. And the last one?
And finally, chemoarchitecture uses advanced immunohistochemistry to tag specific neurotransmitters or their synthesizing enzymes. It shows us not just where the cells are or where their wires go, but exactly which chemical language they are speaking in which specific regions.
And the clinical meaning jumps right out of these architectural maps, if you know what to look for. The textbook uses Alzheimer's disease as a primary example, which the DSM-5 categorizes as major neurocognitive disorder due to Alzheimer's type.
Yes, a very clear example.
Let's use cytoarchitecture to understand it. If you look at a Nissl stain of a specific brain region called the entorhinal cortex in a healthy adult, layer 2 has these beautiful, distinct islands or clusters of very large neurons.
Right.
But if you look at the exact same cytoarchitecture in a brain from someone who died with severe Alzheimer's,
those layer 2 islands are completely devastated. They are gone. Those specific large neurons are uniquely vulnerable to the degenerative proteins of Alzheimer's amyloid plaques and tau tangles.
they just get wiped out.
Completely. Their loss physically destroys the cytoarchitecture of that specific layer. Now, to connect this to clinical meaning, why does that matter? Because the entorhinal cortex acts as the major anatomical interface, the gateway between the hippocampus, which forms memories, and the neocortex, where long-term memories are stored.
So if layer 2 is destroyed, the bridge is out.
Exactly. The specific cellular destruction breaks the circuit required for consolidating new episodic memories. And what is the hallmark defining clinical manifestation of early Alzheimer's disease?
The inability to form new memories.
Spot on.
It's a chillingly clear link between mechanism, anatomical manifestation, and clinical reality. Okay, let's zoom out slightly from individual glia and stains and look at the cortex itself. You mentioned the neocortex has 6 distinct layers.
Yes.
And within these 6 layers, the 2 main players, the 2 broad categories of neurons, are the pyramidal neurons and the non-pyramidal neurons. Let's unpack this metropolis, starting with the pyramidal neurons, which make up the vast majority, about 75% of the neocortex.
Pyramidal neurons are the heavy lifters. They are the primary excitatory projection neurons of the cortex. Excitatory means they use glutamate to tell other cells to fire, and projection means they send their axons across different cortical layers over to the opposite hemisphere of the brain, or all the way down into the brainstem and spinal cord.
Right.
Structurally, they are named for their prominent pyramid-shaped cell body. Out of the top of this pyramid, a single, thick, apical dendrite reaches straight up towards the surface of the cortex. And out of the base, multiple basilar dendrites spread out horizontally.
And these dendrites, the branches, are covered in tiny microscopic protrusions called dendritic spines. If the dendrite is the branch of a tree, the spines are the individual leaves.
That's a great visual.
These spines are the primary recipient zones for excitatory synapses. They are the exact locations where the pyramidal neuron listens to incoming glutamate signals from other cells.
They are. And this brings us right back to our gardeners, the microglia, and the concept of synaptic pruning. Remember we said that in adolescence, microglia prune away unused spines?
Yes, the chainsaws.
Exactly. Well, in schizophrenia, one of the most consistent, highly replicated findings in postmortem anatomical studies is a significant reduction in the number of dendritic spines, specifically on the basilar dendrites of pyramidal neurons.
Everywhere in the brain.
No, and not just anywhere, specifically in deep layer 3 of the dorsolateral prefrontal cortex, or DLPFC.
Okay, let's follow the logic here. The DLPFC is crucial for working memory and executive function, holding information in your mind, making complex decisions.
Correct.
If there are fewer dendritic spines on the pyramidal cells in this area, it means there are fewer excitatory synapses. Those pyramidal neurons are simply not receiving enough stimulation. They're cut off. But why? What causes the spine loss in the first place? Do the microglia just go rogue?
That is the multi-million-dollar research question. The mechanism appears to be a complex, tragic combination of factors. First, there may be a genetic liability affecting something called actin dynamics.
Actin.
Yeah, actin is a protein that forms the internal structural scaffolding inside the spine. Think of actin like the tent poles holding up a canvas tent. If the genes coding for actin are mutated or dysregulated, the tent poles are weak, the spines become structurally unstable.
So you have weakened spines, and then the gardeners arrive.
Precisely. Combine this structural instability with microglia that may be primed to be overactive due to early life inflammation or genetic factors. During late adolescence and early adulthood, which perfectly mirrors the classic age of onset for schizophrenia, the microglia aggressively prune these weakened spines.
Because they look damaged to the microglia.
Exactly. You end up with an impoverished excitatory network. Less excitation means less output from these pyramidal neurons, leading to lower overall cortical activity in the DLPFC. Clinically, this directly correlates with the severe working memory deficits and negative symptoms seen in patients with schizophrenia.
So that is the excitatory side, the 75%. Now let's look at the remaining 25% of the cortex, the non-pyramidal neurons. These are those local circuit interneurons we mentioned earlier, and they are mostly inhibitory, meaning they use GABA to tell other cells to stop firing.
Right.
And they have fantastic, highly descriptive names, like chandelier cells and basket cell.
They do. And while they are fewer in number compared to the pyramidal cells, their functional role is absolutely paramount. If pyramidal cells are the engine, the interneurons are the steering wheel and the brakes. They dictate the timing, the rhythm, and the boundaries of the pyramidal neurons firing.
Let's look closely at chandelier cells.
Okay, chandelier cells are totally unique because their axon terminals form synapses exclusively on a very specific part of the pyramidal neuron called the axon initial segment.
Why is the axon initial segment so important?
Because that is the physiological trigger zone. It's the exact spot where the pyramidal neuron computes all the incoming excitatory and inhibitory signals and makes the final decision on whether or not to fire an action potential down its own axon.
Oh, I see.
So by placing its inhibitory synapses right on the trigger zone, a single chandelier cell has enormous veto power over the output of hundreds of pyramidal neurons. It can shut them down instantly.
And in schizophrenia, this crucial inhibitory system is also struggling, isn't it?
It's deeply compromised. Post-mortem anatomical studies reveal a fascinating pathology. The axon terminals of these chandelier cells in schizophrenic brains have significantly reduced levels of a protein called gate1.
What does gate1 do?
Gate1 is basically the molecular vacuum cleaner. It's the membrane transporter that pulls GABA back into the chandelier cell after it has been released into the synapse so it can be recycled.
So the vacuum cleaner is broken. But wait, concurrently, the pyramidal neurons they synapse onto the receiving cells show an increase in a specific type of GABA receptor, the GABA alpha 2 receptor.
Yes.
I want to pause here and apply some clinical reasoning. If I just read that, it looks like two completely separate random problems. Less GAT1 here, more receptors there. But if we think about the brain as a dynamic living system, well, this doesn't look like random damage. This looks like the brain desperately trying to fix itself.
That is a phenomenal insight, and you are employing a crucial clinical reasoning framework here. When we see anatomical changes, we must determine if they're the primary cause of the disease or a compensation. In this case, it strongly represents compensation. The primary pathophysiological issue seems to be that the chandelier cells are failing to produce and release enough GABA in the first place. The inhibitory signal is weak.
So the brain reacts.
Exactly. The reduction in the GAT1 transporter, the vacuum cleaner, is likely a compensatory mechanism. The chandelier cell is trying to leave whatever tiny amount of GABA it does release in the synaptic cleft for as long as possible to maximize its effect.
I'm trying to stretch out the signal.
Right. Meanwhile, the postsynaptic pyramidal neuron upregulates or increases its GABA receptors. It's essentially becoming hypersensitive, desperately turning up its own volume dial trying to hear the weakened inhibitory signal. It is a struggling, highly altered compensatory network that ultimately fails to maintain proper cortical balance.
And when that balance fails, we lose the rhythm of the city, we lose the synchronized firing. When you hear about brain waves on an EEG theta alpha gamma waves, how do these connect to the cellular dance we're discussing between pyramidal and interneurons?
Well, cortical oscillations brainwaves are generated directly by networks of GABAergic interneurons pacing the firing of the excitatory pyramidal cells. Let's focus on the parvalbumin-containing basket cells, another type of interneuron.
Okay.
Imagine these interneurons as the conductor of a massive symphony orchestra, ensuring all the pyramidal musicians play their notes in perfect time together. Gamma oscillations, which are very high-frequency waves between 30 and 80 hertz, are crucial for integrating information across widely distributed neuronal networks.
So they tie it all together.
They do. They are the literal neural substrate for focused attention, working memory, and conscious perception.
So when you are concentrating really hard, like trying to hold a phone number in your head while simultaneously doing mental maths, your prefrontal cortex is just humming with synchronized gamma oscillations.
Precisely. But in schizophrenia, because of those severe deficits in the GABAergic interneurons we just discussed, the conductor is completely out of sync. The timing is off.
The musicians are just playing whatever.
Basically, yeah. Electroencephalogram, or EEG, studies show that individuals with schizophrenia have significant deficits in the power and synchrony of prefrontal gamma oscillations during demanding cognitive tasks. The underlying circuitry simply cannot synchronize effectively to bind the information together, which directly, mechanically manifests as the profound cognitive dysfunction we see in the clinic.
Okay, we have built the cells, we have looked at the local circuits of excitation and inhibition, and we understand their rhythms. Now let's zoom all the way out. Let's look at the large-scale associational networks.
The big picture.
Exactly. If functional neuroanatomy is a map of a city, these networks are the massive superhighways connecting completely distant boroughs. There are many networks in the brain, but three great cortical networks dominate the modern conversation in psychiatry: the central executive network, the default mode network, and the salience network.
Yes, the big three.
Let's break these down one by one. First, the central executive network.
The central executive network is your task-positive, outward-facing network. Anatomically, it heavily involves the dorsolateral prefrontal cortex and the posterior parietal cortex. This network comes online when you are actively engaged in demanding, externally directed cognitive tasks.
Like what?
Sustained attention, complex problem solving, manipulating data in your working memory, making difficult decisions. It is the network you, the listener, are ideally using right this very second to follow and parse this complex biological discussion.
Contrast that task-positive state with the default mode network, or DMN.
The DMN is the task-negative network. It is formed primarily by the medial prefrontal cortex, the posterior cingulate cortex, and medial areas of the posterior parietal cortex. It does the exact opposite of the central executive.
So it's active when we're not focusing.
Exactly. It becomes highly active when you are at rest, when you are not focused on the external environment. It drives your internal cognition, daydreaming, recalling autobiographical memories from your childhood, envisioning your future plans, and something called theory of mind.
Let's define theory of mind briefly.
Theory of mind is the cognitive ability to attribute mental states, beliefs, intents, desires, emotions to yourself, and to understand that others have beliefs and intentions that are different from your own. It's empathy and social calculation. All of that internal self-referential processing lives in the default mode network.
This is where I want to push back a bit, or at least ask for the exact mechanism, because experience tells me you cannot be intensely focused on a complex maths puzzle and simultaneously be vividly daydreaming about your upcoming holiday to Spain.
No, you really can't.
The brain has to switch between these two networks. They seem mutually exclusive. How does it physically do that?
You're entirely correct. They are generally anti-correlated. When one is up, the other is down. That switching mechanism is the specific domain of the third great network, the salience network, comprised primarily of the anterior cingulate cortex, the ventromedial prefrontal cortex, and the insula. The salience network is your brain's radar. It is constantly monitoring the external environment and your internal visceral state for highly relevant or salient stimuli.
So it's looking for what's important.
Exactly. When it detects something important, a loud noise, a sudden pain, a specific name spoken in a crowded room, it acts as the switch operator. It rapidly deactivates the default mode network and brings the central executive network fully online to deal with the salient stimulus.
Wait, what actually flips that switch at the cellular level? How can a signal move that fast across the entire brain?
It appears to rely heavily on a highly unique type of cell called the von Economo neuron.
Von Economo?
Yes. These are very large, elongated, spindle-shaped projection neurons. What's fascinating from an evolutionary perspective is that they are found almost exclusively in humans and a select few other highly social, intelligent species, like great apes, whales, and elephants.
Oh, wow.
In humans, they are clustered densely and specifically in the frontoinsular cortex, a key node of the salience network. Because they are so large, their thick axons allow for incredibly rapid electrical relay of information across vast brain distances, making them perfectly suited to drive this instantaneous, large-scale switching between global networks.
So what happens when these massive networks become dysregulated? What is the clinical meaning here for a psychiatrist? Let's look at major depressive disorder first.
In major depression, functional neuroimaging consistently shows that the default mode network is massively overactive, and its functional connectivity, how tightly the nodes communicate, is abnormally high.
So they're stuck in internal thought.
Exactly. The patient essentially gets stuck in this internal, self-referential mode of cognition. Clinically, we see this manifest as severe rumination, the repetitive, intrusive, inescapable focus on negative past events, guilt, or perceived personal flaws. Concurrently, because the DMN is hyperactive, connectivity within the outward-facing central executive network is reduced.
Which perfectly explains why severely depressed patients experience profound difficulties with basic concentration, working memory, and decision-making at work. Those switches jammed, and they are trapped in the negative internal task-negative state.
That's spot on.
What about in schizophrenia? How do network dynamics explain psychosis?
In schizophrenia, the pathological issue seems to center heavily on a broken salience network. There's a general breakdown in functional connectivity across all three networks. But a dysfunctional salience network, perhaps mechanically driven by observed gray matter loss in the insula and anterior cingulate, fails to appropriately assign salience or importance to incoming stimuli.
So the radar is broken.
Utterly broken. It misinterprets completely irrelevant external events, like a random stranger glancing at them on the bus or a car license plate, as being highly personally significant. Simultaneously, it blurs the boundary between internal thoughts generated by the DMN and actual external reality.
Hold on. So an internal self-generated thought from the default mode network might be erroneously tagged by the broken salience network as a signal originating from the external world.
Exactly. And that exact anatomical mechanism gives rise to the most distressing clinical manifestations of schizophrenia: psychosis. A delusion is fundamentally a false assignment of profound salience to an irrelevant event.
And a hallucination.
A hallucination is simply an internal thought or an internally generated sensory memory that the brain mischaracterizes as an external sensory perception because the network failed to tag its correct origin.
That is such a powerful way to understand psychosis. It removes the mysticism and grounds it in biology. Now, before we move off the cortex entirely, I want to take a fascinating slight detour.
Sure.
We've talked about these massive distributed networks, but what integrates them? I mean, you have a visual cortex, an auditory cortex, an emotional limbic system, what, Mel? But you experience life as a single, unified conscious moment. The source text mentions a deeply mysterious structure called the claustrum. What is it?
The claustrum is a thin, irregular, almost sheet-like layer of gray matter located deep inside the brain, just deep to the insular cortex, hidden away next to the putamen of the basal ganglia.
Okay, so it's buried in there.
Yeah, deeply buried. Despite its incredibly small size, tracing studies reveal that it has dense reciprocal connections with almost every single region of the cerebral cortex. Because of this unparalleled whole-brain connectivity, Francis Crick, the co-discoverer of DNA, famously hypothesized that the claustrum acts as the ultimate conductor of consciousness.
The conductor of consciousness. That's a huge title.
It is. It supposedly binds sensory, cognitive, and emotional information from disparate, widespread sources into one coherent, unified conscious experience.
And here's the truly wild part for modern psychiatry. The claustrum contains the highest density of serotonin 2A receptors in the entire human brain.
Which is highly, highly significant because the serotonin 2A receptor is the primary pharmacological target for classical psychedelic substances like psilocybin, LSD, and mescaline.
So when someone takes a psychedelic, the drug is basically hammering the claustrum.
It is intensely activating those receptors. And we know that activation of 5-HT2A receptors by psychedelics profoundly alters consciousness, warps perception, and radically shifts global network connectivity, often completely dissolving the rigid boundaries between the default mode and central executive networks, which users describe as ego dissolution.
Right, that feeling of oneness.
Exactly. Understanding the unique anatomy of the claustrum is currently providing a strong biological rationale for the immense, rapidly growing interest in using psychedelics as targeted, network resetting treatments for severe refractory psychiatric disorders like PTSD and treatment-resistant depression.
Okay, we have covered a tremendous amount of ground in the cortex. Now let's dive deeper into the brain, beneath the cortical mantle, and look at the subcortical structures that feed information into the cortex, starting with the thalamus.
The thalamus, yes.
Now, most people learn in basic biology or early medical school that the thalamus is just the brain's sensory relay station. You know, all sensory info except smell stops at the thalamus before going to the cortex. But calling it just a relay is a massive, almost insulting understatement, isn't it?
It's a severe oversimplification that hampers clinical understanding, to be honest. The thalamus is not a single lump. It is a massive, highly complex collection of dozens of distinct nuclei that meticulously regulate what the cortex gets to see, hear, and do.
How do we break them down?
We can broadly divide these nuclei into specific relay nuclei, association relay nuclei, and diffuse projection nuclei.
The specific relay nuclei are what we usually think of. The lateral geniculate nucleus relays raw vision from the eyes to the occipital lobe. The medial geniculate nucleus relays raw hearing from the ears to the temporal lobe. But the association relay nuclei are where it gets psychiatrically complex.
Yeah.
Let's focus on one, the medial dorsal nucleus.
The medial dorsal nucleus, or MDN, is absolutely critical for psychiatry. Unlike the specific relays, the MDN does not receive raw sensory input from the outside world. Instead, it receives highly processed emotional, autonomic, and homeostatic information from internal structures like the amygdala and the hypothalamus.
Okay.
It then projects directly and reciprocally to the prefrontal cortex.
So it's feeding emotional data to our logical center.
Exactly. It is essentially the anatomical gateway that allows our basic emotional drives, fears, and survival instincts to influence our highest cognitive decision-making.
Is that important?
The connection is so vital that if you surgically lesion the medial dorsal nucleus in primates, they develop the exact same severe spatial working memory deficits and executive dysfunction as if you had physically destroyed their prefrontal cortex itself. The cognitive circuit is entirely interdependent on this thalamic input.
And regulating all of this massive thalamic traffic, the sensory relays, the emotional relays, is a very special, incredibly thin structure, the reticular nucleus of the thalamus.
Yes. The reticular nucleus forms a very thin, shell-like sheet of neurons covering the lateral aspect of the entire thalamus. It is entirely unique structurally and functionally because it is the only thalamic nucleus comprised entirely of inhibitory GABAergic neurons.
Oh, the only one.
The only one. Here's how it works. As all the millions of axons travel upwards from the thalamus to the cortex, and as the reciprocal axons travel back downwards from the cortex to the thalamus, they all have to pierce through this reticular shell. As they pass through, they send tiny collateral branches that synapse onto the reticular nucleus.
So it is constantly sampling all the traffic going both directions. It knows exactly what the thalamus is saying to the cortex and what the cortex is demanding to the thalamus.
Precisely. It samples the global traffic, and then, because it is inhibitory, it sends projections back into the specific underlying thalamic nuclei. It acts as the ultimate gatekeeper. It dynamically regulates thalamic output.
Give me an example of how that works in real time.
Okay, if you need to focus on reading a book in a noisy cafe, the cortex tells the reticular nucleus to inhibit the medial geniculate nucleus, which is the auditory relay, suppressing the background noise while allowing the visual relay to pass unimpeded. It shifts our attention by actively suppressing irrelevant information and enhancing what is currently important.
Let's move from the thalamus to another major, incredibly important subcortical system, the basal ganglia. Now, when you hear basal ganglia, which includes structures like the caudate, the putamen, the globus pallidus, the substantia nigra, you immediately think of movement.
You do, yeah.
You think of neurology. You picture the resting tremor of Parkinson's disease or the writhing movements of Huntington's chorea. Why on earth are we spending so much time on the basal ganglia in a textbook about psychiatry?
It's a vital question. We study it because the exact same complex anatomical circuitry that regulates smooth, purposeful physical movement also regulates smooth, purposeful thought, reward processing, and emotion. The basal ganglia are essentially a massive, incredibly complex feedback loop for the cerebral cortex.
Okay, walk me through the loop.
The cortex sends an idea, whether it is an idea to physically move your arm to pick up a coffee cup, or an abstract idea to solve a crossword puzzle down into the input station of the basal ganglia, which is the striatum.
Right.
The basal ganglia process this idea, refine it, and send the refined signal through the thalamus back up to the exact region of the cortex that initiated it. This massive system forms distinct, parallel, topographically organized loops.
So we have motor loops for movement, yes, but we also have cognitive loops and limbic loops for thought and emotion.
Exactly. The motor cortex projects to the putamen, forming the classic motor loop. But the dorsolateral prefrontal cortex, our logic and working memory center projects, the central head of the caudate nucleus, forming a distinct parallel cognitive loop. And the orbitofrontal cortex and anterior singulate our emotional and impulse control cortices project to the ventral striatum, forming the limbic loop.
Okay, to understand how psychiatric illnesses hijack these loops, we need to deeply understand their mechanism. And the basal ganglia operate on a somewhat confusing principle called disinhibition.
Yes. Disinhibition.
But I have to be honest, I always struggle with this. When I want to move my arm, I just assume my brain sends an accelerator signal. But that's not how the basal ganglia works. It's like taking your foot off a double brake system. Can you walk me through this step by step? Break it down for me.
It is highly counterintuitive, but the double brake analogy is the best way to visualize it. Let's trace the main direct pathway. Imagine you are parked on a steep hill. The internal segment of the globus pallidus, or GPI, is your parking brake.
Okay, GPI is the parking brake.
In a resting state, the GPI uses GABA to constantly, relentlessly inhibit the thalamus. It holds the thalamus down, preventing it from sending signals to the cortex.
Okay, so the parking brake is firmly on, holding the car, which is the thalamus, in place. How do we move?
To move, or to execute a thought, The cerebral cortex uses excitatory glutamate to stimulate the striatum. The striatum is your foot brake. The excited striatum then sends a strong inhibitory GABA signal directly to the GPI.
Wait, so the striatum inhibits the GPI, which was inhibiting the thalamus.
Exactly. You are inhibiting the inhibitor. By stepping on the foot brake, the striatum, you release the parking brake, the GPI. Because the GPI is now shut down, it stops inhibiting the thalamus.
And the car rolls forward.
Right. The thalamus is finally released or disinhibited, allowing it to fire its excitatory glutamate signals freely back up to the cortex, successfully executing the desired action or thought. It is an elegant back-to-back inhibitory control system that prevents our brain from firing randomly.
Okay, that actually makes sense. Inhibiting the inhibitor allows the signal to pass. And the clinical meaning here is profound across multiple psychiatric domains, depending entirely on which specific loop is experiencing a brake failure.
Truly profound. Let's look at the cognitive loop involving the DLPFC and the caudate. Dysfunction in this specific circuitry, particularly altered dopamine modulation of the striatum, is heavily implicated in the severe working memory, planning, and executive deficits we see in schizophrenia. The cognitive brakes are faulty.
Makes sense.
Then, if we look at the lateral orbitofrontal loop...
So the orbitofrontal cortex handles impulse control and error detection, right?
Yes. Hyperactivity in this specific closed circuit, where the loop is firing relentlessly, unable to apply the brakes, drives the repetitive, intrusive, anxiety-provoking thoughts, the obsessions, and the irresistible ritualistic physical behaviors, the compulsions, that are the hallmark of obsessive-compulsive disorder.
And what about the limbic loop? That one passes through the ventral striatum, which importantly includes the nucleus accumbens. That's the brain's core reward center.
Exactly. The ventral striatum receives incredibly dense dopaminergic input from the ventral tegmental area in the midbrain. This loop processes reward, motivation, and pleasure.
So when that breaks...
Dysfunction in this limbic loop, a mechanical failure of reward processing, is the biological bedrock of two massive psychiatric domains. First, major depressive disorder, where underactivity manifests as anhedonia, the total inability to feel pleasure or motivation.
And the second?
And second, substance use disorders and addiction, where this exact circuitry is artificially hijacked, flooded, and eventually downregulated by massive spikes of dopamine caused by exogenous chemicals like cocaine or heroin.
It is genuinely incredible how the exact same underlying mechanical principle, the double brake system of the basal ganglia loops, can manifest clinically as a shaking physical movement tremor, a paranoid cognitive delusion, a compulsive hand-washing ritual, or a devastating heroin addiction, purely depending on which parallel anatomical loop is affected.
It perfectly illustrates why a deep understanding of functional neuroanatomy is not just an academic exercise for a psychiatrist. It dictates how we conceptualize, categorize, and eventually biologically treat these illnesses.
Let's continue integrating these vast systems by moving downwards, looking at the limbic system and the brainstem. We have mentioned the limbic system several times, but what exactly is it? Because it seems like every textbook draws the boundaries slightly differently.
You're right. The term has a rich and slightly controversial anatomical history. In the 19th century, the French neurologist Paul Broca identified the limbic lobe, from the Latin word limbus, meaning border or margin, as the distinct rim of cortex encircling the diencephalon.
The inner rim.
Yes. Then, in the 1930s, James Papez proposed a specific, closed neural circuit connecting the hippocampus, the thalamus, and the cingulate cortex as the anatomical basis of human emotion. Later, Paul MacLean expanded this idea into the broader limbic system, explicitly incorporating structures like the amygdala and the hypothalamus.
But it's not a strict geographical border today, right?
No, today, strict anatomists argue endlessly about its exact boundaries, mainly because its structures are so heavily and seamlessly integrated with the rest of the brain. But conceptually, for the clinician, it remains a vital, indispensable framework for understanding how we generate and regulate emotion, how we form emotionally charged memories, and how we intimately link higher cognitive processing with our primitive autonomic survival drives.
Let's isolate a few key structures within this conceptual system. The cingulate gyrus is a massive part of this inner rim. And specifically, I want to look at the subgenual anterior cingulate cortex, famously known as Brodmann Area 25.
Brodmann Area 25 is located ventrally, right down on the midline, just below the knee of the corpus callosum. Anatomically, it has highly extensive bidirectional connections to the amygdala, the hypothalamus, and the autonomic centers in the brainstem.
And clinically?
As we discussed earlier regarding networks, functional PET and fMRI imaging reveal that this specific cortical area is pathologically overactive, burning bright red on a scan in individuals suffering from severe treatment-resistant depression.
And the clinical application of that specific anatomical knowledge has been nothing short of revolutionary in recent years.
It has been a paradigm shift. When severely depressed patients do not respond to multiple trials of antidepressants, intensive psychotherapy, or even electroconvulsive therapy, we can now use deep brain stimulation, or DBS.
That's the pacemaker for the brain, effectively.
Exactly. By surgically implanting a microscopic electrode deep into the brain to deliver continuous high-frequency electrical stimulation directly to Brodmann area 25, we can forcibly downregulate its pathological hyperactivity. It is effectively using a targeted electrical pacemaker to physically reset a dysregulated mood circuit. And in many cases of intractable profound depression, the clinical results are startlingly effective.
Another tiny limbic structure that is currently gaining a massive amount of attention in mood regulation research is the habenula.
The habenula is structurally tiny, located dorsally in the epithalamus, but it acts as a massive, highly influential central hub. The lateral portion of the habenula connects the limbic system and the basal ganglia directly down to the deep brainstem centers that actually produce our primary neurotransmitters. The raphe-nuclei, which produce serotonin, and the ventral tegmental area, which produces dopamine.
I've heard the habenula referred to as the brain's disappointment center. How does that work?
It's an apt nickname, really. In animal models, we see that when an expected reward does not happen, when an animal expects a drop of juice but gets nothing, the lateral habenula fires incredibly strongly.
That complains.
Basically. And when it fires, it sends inhibitory signals that actively shut down the dopamine and serotonin systems. It literally broadcasts disappointment to the brain. In severe depression, research suggests the habenula becomes pathologically overactive, constantly inhibiting reward pathways. This chronic overactivity drives the profound anhedonia and pervasive negative mood, making the habenula another highly promising emerging target for targeted neuromodulation.
And we absolutely cannot discuss the limbic system without dedicating time to the amygdala. For decades, pop science and early textbooks have branded the amygdala simply as the brain's primitive fear center. But calling it a fear center is way too simplistic, isn't it?
Far too simplistic. The amygdala isn't just a single blob. It is a complex composed of several distinct anatomically separate nuclei. The largest of these is the basolateral complex.
What's special about it?
What is fascinating about the basolateral complex is that if you look at it under a microscope, it does not look like a primitive, unorganized subcortical mass. Its cytoarchitecture and its connectivity patterns are remarkably similar to the advanced cerebral cortex. It has dense, robust, bidirectional connections with the prefrontal cortex and the medial dorsal thalamus.
So it's not just a primitive alarm bell blindly triggering panic. It is actively integrating our raw emotional state with our highest cognitive reasoning.
Precisely. It actively evaluates the emotional salience of incoming sensory input and constantly cross-references it with our cognitive goals and past memories. And when it does need to initiate a bodily response to a threat, it doesn't just send one signal, it uses two massive, distinct output highways.
What are they?
First, the sestria terminalis, which arches dorsally over the thalamus to hit the hypothalamus, and second, the ventral amygdalofugal pathway, which dives ventrally straight down into the brainstem and the thalamus.
Why two pathways? It seems redundant.
Redundancy and breadth of response. This dual routing ensures that an emotional stimulus like seeing a snake simultaneously triggers an immediate unconscious autonomic physical response via the hypothalamus, spiking your heart rate,
Right.
while also triggering a conscious cognitive appraisal via the thalamus and prefrontal cortex, allowing you to realize it's actually just a garden hose.
Which brings us perfectly down to the absolute roots of the mind, the brainstem. We tend to think of the brainstem, the midbrain, the pons, and the medulla as just handling the boring vegetative stuff. Breathing, heart rate, swallowing. But it is the absolute foundation of the psychiatric experience. Without it, there is no mind. Let's start with a concept that fundamentally relies on the brainstem, interoception.
Interoception is your brain's continuous, mostly subconscious perception of your body's internal physical state. It's the feeling of your heartbeat, the state of your digestion, the expansion of your lungs, the tension in your gut.
Basically feeling your inside.
Exactly. Sensory nuclei in the brainstem gather all this continuous visceral data from the vagus nerve and project it upwards to the insular cortex. This continuous rhythmic flow of interoceptive data is arguably the biological genesis of your fundamental sense of self. You know you are you and that you inhabit a physical body precisely because you can feel the internal physical boundaries and rhythms of that body.
And in psychiatric disorders, this vital sense of internal self is often deeply distorted.
Deeply so. Let's look at two extremes. In schizophrenia, patients often show significantly reduced interoceptive accuracy. They're neurologically disconnected from their own bodily signals.
So they don't feel their own body states well.
Right. This profound sensory disconnect likely contributes to the fragmented sense of self, the feeling of depersonalization, and potentially the failure to recognize their own internal thoughts as belonging to them.
And the other extreme.
Conversely, in conditions like obsessive-compulsive disorder or panic disorder, we see heightened interoceptive sensibility. The patient's insula is hyper-focused on every minor normal fluctuation in their heartbeat or breathing, instantly interpreting normal physiological noise as a catastrophic, life-threatening event.
Let's move slightly higher in the brainstem to the midbrain. We talked about the dopamine centers here, but there is an area called the periaqueductal gray, or PEG. This structure is absolutely fascinating in the clinical context of trauma and PTSD.
The PEG is a tube of gray matter that surrounds the cerebral aqueduct in the midbrain. It is a major, highly complex relay center for processing pain and coordinating defensive survival behaviors. Anatomically, it is organized into distinct vertical columns, and they do very different things. The dorsal columns of the PEG receive heavy inputs from the amygdala and drive active, sympathetic nervous system responses, the classic fight or flight mechanisms, increased heart rate, blood directed to muscles, aggression, or fleeing. However, the ventral columns of the PEG, conversely, drive parasympathetic passive coping mechanisms.
Passive coping, you mean like playing dead?
Exactly. Freezing, bradycardia, which is the slowing of the heart rate, profound physical immobility, and psychological dissociation.
And how does this specific anatomy relate to the clinical presentation of post-traumatic stress disorder?
In a healthy, non-traumatized brain, the logical prefrontal cortex exerts strong, top-down inhibitory control over the amygdala, keeping its alarm signals in check. In severe PTSD, functional imaging shows that prefrontal control is significantly weakened.
So the amygdala is off the leash.
Exactly. The amygdala becomes hyperreactive. It sends unchecked, massively amplified, inappropriate excitatory signals down into the periaqueductal gray. This anatomical misfiring explains why a completely safe, minor trigger in the present environment, like a car backfiring, can instantly plunge a patient with PTSD into a massive, overwhelming, sympathetic fight-or-flight panic via the dorsal PEG.
Wow.
Or, conversely, it explains why they might suddenly collapse into a profound, unresponsive, dissociative freeze state via the ventral PEG. It is not a psychological weakness. It is a rapid, catastrophic misfiring between the cortex, the amygdala, and the brainstem survival columns.
Finally, just below the midbrain is the pons. And hidden deep within the pons is a tiny, incredibly influential cluster of blue pigmented neurons called the locus coeruleus.
The locus coeruleus is the predominant source of the brain's norepinephrine, also known as noradrenaline. Despite being an incredibly tiny nucleus, only a few tens of thousands of cells, its axons branch out so extensively that they innervate virtually the entire cerebral cortex, the hippocampus, and the thalamus.
It goes everywhere.
It really does. Norepinephrine acts globally as a neuromodulator to instantly increase physiological arousal, alertness, and attention to the external environment.
So it's essentially the brain's global alarm system. When you hear a branch snap in the dark woods, the locus coeruleus floods the cortex with norepinephrine, so you are instantly hyper-focused. But what happens when that system is physically dysregulated?
You see profound, debilitating psychiatric manifestations. Hyperactivity in the locus coeruleus noradrenergic projections is deeply implicated in panic disorder and generalized anxiety disorder. Imagine that tiny alarm bell at the base of your skull.
Right.
In a healthy brain, it rings loudly when a tiger jumps out at you. But in panic disorder, the neurochemical wiring is frayed, and that bell is spontaneously blaring at 120 decibels while the patient is just sitting quietly in traffic or lying in bed. The entire cerebral cortex is flooded with a massive chemical signal of extreme urgency and mortal threat when absolutely no external threat exists. That mismatch is a panic attack.
Okay, we have traced the anatomical pathways from the microscopic astrocytic power grid through the massive cortical networks, down the disinhibitory basal ganglia loops, and deep into the brainstem survival centers. To tie all of this structural knowledge together into applied clinical practice, let's talk about the 5 Cs.
Yes, the 5 Cs.
As a clinician, when you read a new research paper saying, brain scans show significantly reduced volume in the hippocampus of patients with major depression, you cannot just accept that at face value. You have to actively interpret it.
Exactly. An anatomical difference on a scan or slide is just an observation. It is a data point. Clinical reasoning in psychiatry requires us to rigorously determine what that anatomical difference actually represents in the context of the living patient. We use the 5 Cs framework. First is the structural anomaly the cause, meaning, did a genetic malformation of the hippocampus directly trigger the onset of the depressive disease?
Or conversely, is it a consequence?
Right. Perhaps the depression itself or the chronic toxic flood of cortisol associated with the stress of the disease caused a neurotoxic loss of hippocampal tissue over years of untreated illness. The depression caused the brain change, not the other way around.
And then there is the third C, compensation, which we discussed at length with the upregulation of GABA receptors on the pyramidal cells trying to make up for the failing chandelier cells in schizophrenia.
Yes. Compensation. We must ask, is the brain actively altering its structure, its receptor density, or its network connectivity, not because it is inherently damaged, but as a desperate homeostatic attempt to fix a primary failure elsewhere in the circuit?
The fourth C is comorbidity. This seems incredibly common in psychiatric research.
It is the absolute bane of psychiatric studies. Is this brain change genuinely related to the primary psychiatric diagnosis of, say, bipolar disorder? Or is it actually due to a comorbid factor that almost always accompanies the illness, like a 30-year history of heavy tobacco use, chronic alcohol abuse, poor metabolic diet, or severely disrupted glymphatic sleep clearance?
And finally, the most frustrating one for researchers, the confound.
The confound. Is the anatomical difference we are observing merely an artifact of our research methodology? In postmortem studies, is the altered cell count simply due to the postmortem interval, how long it took before the tissue was chemically fixed?
Oh, wow, right.
Or, most commonly and problematically in functional neuroanatomy, is the observed structural change-like basal ganglia enlargement actually a physiological side effect of the chronic pharmacological antipsychotic treatment the patient received for 20 years, rather than a symptom of the schizophrenia itself? Mastering the integration of neuroanatomy into clinical practice requires constantly, ruthlessly filtering every single finding through these 5 Cs.
That is a phenomenal, highly practical framework. We are nearing the end of our deep dive into the architecture of the mind, so it is time to distill everything we have discussed into our key takeaways, directly summarizing the mission of Chapter 1.2 of Kaplan and CDOC.
Excellent. Let's run through them.
Here they are. Takeaway 1. Brain function is fundamentally organized across widely interconnected circuits and networks, rather than residing in isolated, static geographical regions.
Yeah, the metropolis, not islands.
Exactly. Takeaway 2. Functional neuroanatomy provides the map that links physical biological structure to complex human domains, such as emotion, cognition, and behavior. Takeaway 3. The neocortex, the limbic system, the basal ganglia, and the primitive brainstem do not operate alone. They operate as tightly integrated, interdependent systems,
all talking to each other constantly.
Takeaway 4. Psychiatric disorders overwhelmingly reflect dynamic dysregulation within these circuits, such as a failure of top-down frontal limbic balance in PTSD or broken disinhibitory frontostriatal loops in OCD. Takeaway 5. Strict anatomical localization provides us with vital clues, but it is the connectivity between regions that explains the true complexity of the mind.
The wiring is what matters.
Takeaway 6. Clinical reasoning in modern psychiatry absolutely involves mapping a patient's symptoms to their underlying neural systems and critically evaluating any observed changes using the 5 Cs. And finally, takeaway 7. Understanding dynamic, living networks is infinitely more useful for a practicing clinician than merely memorizing a static list of isolated anatomical structures.
Those are the core pillars of the chapter. If you can keep those principles central in your mind, the immense, sometimes overwhelming complexity of the human brain becomes a navigable, highly logical clinical landscape.
To really cement this knowledge for you, the listener, we are going to do some active learning reinforcement. I have 6 single best answer, or SBA questions modeled after board exams. I will read the question. I want you to actively pause the audio, commit to an answer in your head, and then hit play to hear the expert breakdown. Ready? Let's begin. Question 1. Which specific glial cell type is primarily responsible for creating the tripartite synapse and actively regulating both glutamatergic and GABAergic neurotransmission in the gray matter? Pause for a moment.
Right. The correct answer is protoplasmic astrocytes. Remember the distinctions here. Fibrous astrocytes are mostly in the white matter for structural support, and microglia are the immune-derived gardeners responsible for pruning. But it is the bushy protoplasmic astrocytes in the gray matter that form that crucial neurovascular bridge to supply energy and intimately wrap around the synaptic cleft to actively clear excess glutamate and prevent excitotoxicity.
Brilliant. Question 2. A young adult patient with schizophrenia exhibits severe working memory and executive function deficits. Postmortem cytoarchitectural studies of their dorsolateral prefrontal cortex are most likely to show a significant reduction in what specific neuronal structure? Pause now.
The answer is dendritic spines, specifically on the basilar dendrites of deep layer 3 pyramidal neurons. The pyramidal neurons are the main excitatory drivers of the cortex, and they receive their vital input on these tiny spines. A loss of spines, perhaps due to genetic actin instability combined with aggressive overpruning by microglia, leads to less overall excitation, less cortical output, and the direct mechanical manifestation of working memory deficits.
Excellent. Question 3. Which large-scale cortical network is considered the vital switch between internally directed thought and externally directed attention, and what specific, uniquely human brain cell type is heavily implicated in facilitating this rapid switching? Pause now.
So the answer is the salience network, and the highly specific cell type is the von Economo neuron. The salience network detects relevant stimuli and toggles the brain between the internal default mode network and the task-focused central executive network. It relies heavily on the massive, rapid transmission capabilities of the spindle-shaped von Economo neurons located in the frontoinsular cortex.
Question 4. Which highly unique nucleus of the thalamus acts exclusively as an inhibitory modulator, sampling both cortical afferent and efferent activity in order to dynamically regulate thalamic output to the cortex? Pause now.
That would be the reticular nucleus of the thalamus. It is anatomically entirely unique because it is the only thalamic nucleus composed entirely of inhibitory GABAergic neurons. It sits as a thin shell around the thalamus, constantly sampling the two-way traffic between the thalamus and cortex, and sends inhibitory signals back down to specific thalamic relays to selectively gate the flow of information.
Question 5. In the modern surgical treatment of severe refractory treatment-resistant depression, deep brain stimulation is sometimes used to effectively downregulate a pathologically overactive cortical limbic area. Which specific anatomical region is typically targeted by the electrodes? Pause now.
The targeted region is the subgenual anterior cingulate cortex, famously known as Brodmann Area 25. Functional PET and fMRI imaging consistently show pathological hyperactivity in this specific node of the limbic circuit in severely depressed patients, making it a prime, highly effective target for electrical neuromodulation to reset the mood circuit.
And finally, question 6. When critically evaluating an altered brain structure in a psychiatric patient, such as altered receptor densities, a clinician must determine if the change is a deleterious, toxic effect of the disease itself or the brain's homeostatic attempt to restore network balance. In the 5 C's framework of clinical reasoning, what does the latter scenario represent? Pause now.
That scenario clearly represents compensation. The human brain is not a static machine. It is highly dynamic and plastic. Alterations like the upregulation of GABA receptors seen on the pyramidal cells in schizophrenia often represent the brain desperately adapting its own architecture to compensate for a primary failure-like failing chandelier cells elsewhere in the network.
Spot on. As we wrap up this extensive deep dive into the architecture of thought, I want to leave you, the listener, with the final provocative clinical thought to mull over. We began this hour by comparing the human brain to a bustling, highly interconnected metropolis built on complex, shifting, pruning, and compensating networks. If the fundamental architecture of our thoughts, our moods, and our sense of self relies on a living biological infrastructure that is constantly rebuilding and rewiring itself in response to its environment, well then, psychiatric treatment is never just about opening the hood and fixing a broken part.
Absolutely not.
You aren't a mechanic replacing a spark plug. It is about providing the precise biological, pharmacological, and experiential environment necessary for the metropolis to rebuild its own roads. Think deeply about how your specific pharmacological choices and your nuanced psychotherapeutic interventions act jointly as the vital urban planners for that biological rebuild.
Right.
Here are the details of this episode.
Functional neuroanatomy. Byline. The architecture of thought mapping structure to function in the living brain.